Thank you very much. It's really my pleasure. I think I have given talks in every TASSA meeting. And this year I was hoping to escape, but of course no such luck with Haluk in the realm. So I think we started with a very broad picture. I'm going to end this session with a very narrow story, a very simple kind of village science type of a story related to obesity. And so these are some of the themes while you're watching this before I start of the things that I hope to touch upon. I want to really thank my laboratory. I have been very blessed to have the chance to recruit really outstanding people all throughout my career. This year actually is 20th year of my lab, and we'll have a retreat in Turkey. I'm incredibly excited about that. So I'll take all my lab and alumni to Turkey. And I also need to acknowledge our supporters, and particularly the Sabri Care Center, the most recent one, which has given us a great momentum and a long-range ability to think about big projects. And the topic that I'm going to talk about today briefly is a collaborative work with industry. This is a really joint effort with UCB, from discovery to translation. And that's the theme of my talk. So I'm going to pose one question, describe one approach, and then tell you one story about potential translation. So the problem, as Murat mentioned, is chronic metabolic diseases. This is the biggest global health problem right now, beyond anything else that you can imagine, including wars, water problems, climate change. The cost is approaching that scale. Both cost in terms of money, but also human opportunity, human welfare. And just a really staggering number here, a study done by David Bloom at the school suggests that 5 of these chronic diseases, cardiovascular, diabetes, atherosclerosis, pulmonary disease, and cancer, just 5 of them will require half of global GDP in terms of health costs in 25 years. So, of course, there is no such source. This kind of source cannot be allocated. Therefore, hundreds of millions of people, that means, will not be treated. So they will suffer from these diseases and won't have access to health care. So it's really an enormous problem. It could be defined in many different ways. This is the problem that I have been working all throughout my career. So as you can see, this is a cluster of problems. They actually don't come one by one. You get one, you get risk for the other. And some of them we understand very well, such as type 2 diabetes is frequently accompanied by obesity. But some of them we don't really quite understand so well. For example, asthma and airway disease. So what does obesity have anything to do with airway disease? And also cancer. For example, right now you heard from Sardar that obesity is the major driver of cancer in terms of mass numbers, although the risk per individual is not very high. The total burden is enormous. So the question that I want to pose today is this. So why do people develop these diseases, especially diabetes? So the example I used was diabetes. But more importantly, why do they develop it so often? I mean, if you wait long enough and wait for the entire world to change its face, almost everyone will suffer from these diseases. It's almost an accelerated version of aging. And so why is that? Why can't something be so frequent? And if it is so frequent, why do we call it disease? Maybe this is the normal state, and the healthy ones are actually that we should be worried about. And this is actually a picture from one of my former fellows who's now a professor in Iowa. It's a picture of a liver cell from an obese individual, which actually turns into a bit of a scary character. So speaking of frequency, of course, if something is more frequent, that means that there has to be a very strong evolutionary reason for that. And so the argument I will make is high blood glucose, so which is the definition of diabetes, is of no concern to evolution. So evolution has really did not develop anything to combat this condition, because if you develop this, then you're disposable for evolution for a long time. The real concern during evolution was the opposite, actually, the low glucose or hypoglycemia, because this is a real threat for survival. For someone, even if not treated to die from diabetes and depleted from the population or the breeding stock will take tens of years, whereas hypoglycemia can kill you in 2 seconds. So therefore, the threat to survival is hypoglycemia, and as a consequence of that, powerful mechanisms, evolutionary mechanisms, must have evolved to prevent a drop in blood glucose during starvation. So everything, actually, this concept really is the basis, in my view, of chronic metabolic diseases. Because there is no such thing as obesity during evolution. Evolutionary forces don't care about this. If you get obese, you get slow, you become delicious, the predators will eat you. And therefore, this is not an issue. The issue is starvation. So if the evolution needs to equip you with things to resist starvation. And therefore, all of this machinery designed to preserve the species actually has now become a liability. And this certainly is true for diabetes. So what are these mechanisms? So these are actually called glucose counter-regulatory hormones. So when your blood glucose drops, there are hormones, first the stress response hormones, and then the pancreatic hormones will actually start stimulating glucose production, and this happens at the liver. So this actually, since the discovery of insulin, this has been the prevailing dogma, that counter-regulatory hormones will start stimulating liver to produce glucose, and then you actually continue to survive. But there's a big missing piece in this since about 100 years. So when you're starving, or when your blood glucose is driven to low, your fat tissue starts breaking its stores, because that's where the energy is stored. And the energy is broken, and these substrates are provided to the body to utilize to pass through this emergency state. But how this adipose tissue really communicates with liver has not been discovered for about, again, a question of 100 years, a century-old question. So most of the people thought that adipose tissue breaks down its lipids, and these lipids come to liver as building blocks to serve for lipid production and glucose production. However, the work from Dennis McGarry, from late Dennis McGarry, unfortunately he passed away, from UT Southwestern, and more recently from Jerry Schulman, showed that this actually idea does not hold up. So the lipids actually do not have anything to do with signaling glucose production. So what is this? And the importance of this, of course, this is the most critical component to really support the survival response. Without understanding this coupling, we really cannot solve this puzzle. And the wishful thinking behind this is, if we understand it, then we could actually intervene with a very critical and central signal. So as Murat mentioned, our really long-term interest resides in the fat tissue, or adipocytes. It's a very uninteresting tissue for many of you, but this is what I see in my dreams every night. Sometimes when I'm in nightmare, in floating and trying to find myself between the fat cells. But again, when I started my lab, or my actually doctoral studies, there was nothing known about this tissue other than it stores energy and it supplies energy at times of need. But in the past 25 years, and some of the work contributors from our group and many hundreds of other groups actually demonstrated that this actually is the most sophisticated endocrine organ in the body, far from the truth that people thought about for 100 years. So this tissue produces many hormones, and then, as mentioned, many of these, interestingly, are immune molecules. So this is actually a very immunometabolic organ that both the adipocytes and immune cells interact to supply the body's hormonal and energy needs. And the question here is the main supply, which is the breakdown of lipids. So this is triacylglycerol. It's broken down to produce free fatty acid and glycerol. And these are the substrates that travel to distant organs. But what is the signal coupled to this process to communicate with the other organs? So our approach was to search for such mechanisms and mediators, and I'll give you one example. This is one of the very first experiments done in my lab 20 years ago. So we were looking for that signal, and we found the lipid binding protein, very simple protein called AP2, which is very heavily regulated in adipocytes while they're forming. So this is early cells, these are mature cells, but interestingly, same in the macrophages. Again, early on, very low. As they become a macrophage, a very critical immune cell, they produce very high levels of this. So I don't really have time to cover the story behind this molecule. It turned out to be a very fascinating molecule. But one thing that always puzzled us, but when we play with this in the adipocyte, which is far from the organs of interest, we saw dramatic effects in the liver. So the question was, how could this happen? How could someone resides in the fat cell control the behavior of liver? And this actually, to tell you how great we are, it took us just 20 years to see something which was sitting right in front of us. And so that question was first tackled by Hyman Chow, who's now an independent investigator at the NIH, Motohiro Sekiya, who's now an independent investigator in Tokyo University. And these are some of our recent members. Furkan is here. He's doing clinical training. Merit was a graduate student. Girol is our long-term colleague. And Karen is doing most of our animal models. And so while tackling this question, so we didn't know. We thought this protein reside here and then send some signal. And Moto, actually, this is an actual picture from the lab. He really spent day and night to try to address this very challenging question. And it turned out that actually this was very simple. It was itself. So itself was actually traveling from adipocyte to liver to do its biology. So Hyman showed this, that adipocyte actually produced this protein. And then most interestingly, when you make obese animals, the levels of this protein went up dramatically. And if you look at the same human version of this animal, the same exact thing happened. This is also a really message for people who think that mouse work doesn't apply to humans. This is the same exact amino acid change. Same exact amino acid change in these two creatures. It produces exactly the same phenotype. And then it turned out that this protein is secreted through a very complicated pathway. Again, please don't look at here, but just you can read this. The exclusive signal for the release of this protein was breakdown of lipids. So it almost was a dream come true, because we found the first secreted hormone from adipocyte that is coupled to breakdown of lipids. And it used a very private secretory pathway, which probably will keep us for the rest of our academic career busy. It doesn't follow the classical secretory pathway that some of you who are educated in biology or medicine read in your textbooks. But once this protein is released, actually the second criteria was what does it do to the liver. So we can do actually an experiment called pancreatic clamp study by labeling glucose. We can measure where the glucose is produced in the body and where does it go in the body in living animals in a real-time manner. So when we did these experiments and gave this protein to normal animals to the level that you see in obesity and diabetes, we see increase in hepatic glucose production. This is a fairly significant, actually, stimulation of hepatic glucose production. And this is the enzymatic machinery to support this. So the first part of our postulate seemed to be adhering that excess production of this molecule from adipocyte stimulates liver to produce more glucose. So coming back to this original model, so if our ideas are halfway true, we might actually have honed into the signal that links adipocytes to liver through this simple protein. So what is the implication of that? So my last part, few minutes, I want to talk about translation, because this is what we promised to talk today. So these are the models that we use. This is actually even a more extreme model. This is the fattest mouse that ever came out of our lab. It weighs 96 grams. It has 4 different genetic mutations. And I'll show you something very fascinating about this creature in few slides. So if the first proposal has validity, so that there are signals which are evolutionarily designed to keep blood glucose high, however, in the age of plenty, remain as a high signal, therefore kept liver producing glucose, causing diabetes, we should be able to interfere with this. So they should be fundamental to metabolic regulation, And perhaps we should be able to introduce ways to intervene with their activity for therapeutic purposes. So this idea was the basis of our collaboration with UCB, which is in its 8th year now. So even the simplest, really, translational project takes many, many, many years to produce anything useful. And we don't know, actually, in this case, whether it will produce anything useful. So before, of course, we do think about medicines to interfere, we have to produce proof of principle. So if we actually remove this gene or block this gene, would that produce any benefit? So this is done very commonly now, and we've done this for many years in the mouse. So we took this gene out of the mouse and then examined their phenotype. So this is actually 20 years of work, but I'm going to show you this, which we haven't published, and I've actually never presented this in anywhere else before coming here, and many of the things I talk about today also. So not only these animals actually became metabolically indestructible, almost no metabolic disease can be induced in these animals, including atherosclerosis that was described to you today. So no matter what you feed to these animals, they never become metabolically ill. They don't develop diabetes. They don't develop cardiovascular disease. They don't die from these diseases. So all of that work is published, but Kenichi Charles, who was a graduate student, a terrific graduate student in the lab, did this experiment to ask a question whether how long this healthy phenotype persists. And she found that actually this persists all throughout life. So they don't live longer, which is a very interesting observation. So we thought they will live forever, but they die exactly at the same time with their brothers and sisters who carry this gene. But until the day they die, they remain healthy. So we don't know actually why they die, and that is a very interesting question all by itself. So if you look at some of the signs, mouse actually develop the same kind of aging signs that we do. You lose hair, the skin got not so vibrant, the hair loses its shine, and none of this actually happens when we delete this gene. So this is what happens to your fat tissue as you age. So this is a young fat tissue filled with juicy fat cells. And then as you age, these cells degenerate, die, and it turns into an inflammatory space, just like a sprained ankle that immune cells infiltrate. You become fatter, as we all experience, a little change in our composition, and then your liver is infiltrated with fat, your blood glucose goes up. So all of these things happen as you age, and none of these things happen in the absence of this gene. For example, the liver just before death is as good as a liver of a 10-week-old animal. The adipose tissue remains healthy and vibrant all throughout their life. So does this have anything to do with humans? So it turns out that this is work done and published by Gurol and Ebru, and recently, actually, we didn't take this work so seriously at the time, but it was reproduced by others. So if you are a lucky, very few lucky individuals have mutations in this gene, and they don't develop heart disease, and they don't develop diabetes. So there is a human proof of principle for this as well. And so most recently, this is Furkan's work just published, and I'm going to show one more slide and finish. So we were able to develop an antibody, and I mean, unfortunately, we gave another very boring name to this antibody called CA33, just like AP2. And so with this antibody, we can reproduce all of the effects of genetic deficiency, at least in the animals. So this actually, here's, for example, one example. If we give this antibody, we can clear their livers, lower their blood glucose, and sugar production from the liver. So this is my final slide. In the middle of this picture, I think we placed one critical hormone that controls the communications between different organs and ensures the healthy contribution of adipocyte into the metabolic health of the body. And whether or not this will turn into a useful application for humans, we'll have to see. But as we speak now, we started primate studies. So this antibody is now given into monkeys. So we'll know for a couple of months whether the efficacy translates further. So I want to end by, again, thanking the Sabri Ilker's support and invite you to our first symposium, which is going to be May 5 and 6. We have a terrific lineup of who is who in metabolic research, and some of the young and brightest stars emerging from the diaspora. Thank you very much.